Okay, good evening. Welcome to the inaugural talk of our new GeCIP and Queen Mary's lecture series. Before we start, can I just ask you all to put your phones on silent so they don't interrupt the speakers if you get a call. I hope you'll all continue to come on the first Thursday of every month for the rest of the series. Last time I was here was in January 2016, listening to Dan MacArthur talking about the GeCIP project, and it seems an extraordinary number of exomes to have as a public reference data set. And I think at the time the 100,000 Genomes Project was really in its infancy, or maybe toddlerhood. There was somewhere between 5,000 and 10,000 genomes, and they're really struggling to extract good quality DNA from tumours. So it seems quite astounding that less than 3 years later we really had no trouble populating this seminar series with really good speakers who are working on the genomes from the 100,000 Genomes Project in our research environment. And not working on a few thousand, but working on tens of thousands of genomes, and some of them the world's largest cancer collections for particular subtypes. And Dr. Will Cross will talk more about that this evening. We're really lucky to have two outstanding speakers this evening. I think that it's not an exaggeration to say that Dr. Will Cross is a pioneer in the 100,000 Genomes Project. He came in early, and that didn't really give him a sort of early starter's advantage. It gave him a lot of problems to deal with, him and his colleagues, and I think anybody working in the research environment now has got a really big thank you to say to all the early onboarders who made the research environment a better place. And Dr. Malak Abed AlThagafi is a special guest speaker this evening. She's also a pioneer in her own national genome project, and we're looking forward to hearing her talk. That's enough from me. I'm going to hand you over to Mark, who doesn't need any introduction. He's our chief scientist, and I think without him and the GeCIP system that he established, we certainly wouldn't be here tonight giving this seminar series, and it would be really very difficult for us to maximise the benefit from the 100,000 Genomes Project and to do big collaborative projects that's really going to allow us to see the potential of the project. So I'll hand over to him. Thank you, Mark.
So just to add my welcome to you all, and thank you for coming. I know how busy you are. We have two fantastic speakers, and it's a pleasure to see you here this evening, and also it'll be a particular pleasure to hear data from the programme. I've just been asked to give you a slight sense of where we've reached in our quest. So we now have, from the National Health Service in England, Scotland, Northern Ireland and Wales, 109,611 samples collected in the NHS by the NHS. And we have generated, this is increasing in the time you're sitting here, it will increase, 91,100 whole genomes sequenced, and they split as you can see there. The bioinformatics team from Genomics England have returned 37,591 reports to the National Health Service. And in rare disease, that's giving a potentially actionable diagnosis in 25%. In some disorders, it's much higher. So certain forms of intellectual disability or developmental disorders were up at between 37 and 40%, and in certain eye diseases between 40 and 50% diagnostic yield. In cancer, we're pleasantly surprised to see potential utility in terms of choosing an agent either available in the National Health Service or a trial opportunity for a patient in perhaps 50% of the participants. But I expect that may go down somewhat as we expand that. And you will see, and I'll give the breakdown in a moment, that the team here from the GeCIP and our research environment team with our research platforms team released 71,860 of those genomes into the research environment yesterday. And there are now 1,800 members who are doing research of the Genomics England Clinical Interpretation Partnership, and we're working with over 70 companies. So here is the data that's available to you today inside our research environment. That is 71,860 genomes. You can see the break between cancer and rare disease, and that's on over 85,000 participants. And we now have secondary data sets alongside the primary data fields, and the clinical data team at Genomics England have done a great job in making those available in a usable format. To give you an idea of how rich that data is becoming, we have secondary data on 85,000 people from NHS Digital. This is hospital episodes, registries, and sadly sometimes death, with cause of death. We've got 7.3 million records on the 85,000 people and 1.6 billion data points. And that's without the primary care data, which I've just been on a call with NHS Digital pursuing. Sadly, 2,141 of the people in the programme have died. That's to be expected, obviously, as it's focused on rare disease and cancer where mortality is high. The 3,000 researchers from 387 academic institutions in more than 24 countries are now volunteered to be part of a coalition of research intellects that drive up the value of this data for clinical care. And the great thing that Anna and the team here and the GeCIP team are seeing is the beginnings of the research environment generating new diagnoses for patients, and often not just for one family or one group of cancer patients, but for many. And so we're particularly seeing that in rare disease. So yesterday at Paul Culler's house, the Minister for Health, Lord O'Shaughnessy, and the Secretary of State for Health announced the inception of a new genomic medicine service, which is what we've been working towards in the 100,000 Genomes Project for the past 5 years. And I could have said 18 months ago I was not entirely sure we would pull this off. But in the first quarter of 2019, at some point, whole genome sequencing will go live for certain rare disease patient categories and cancer in certain subtypes of tumours for over the next 5 years. And so what the Secretary of State has recently announced at the Conservative Conference was that the 100,000 Genomes Project would expand to 1 million funded whole genomes, which will be 500,000 undertaken over the next 5 years by the National Health Service in England, and the 500,000 people from UK Biobank will also undergo whole genome sequencing. But he also announced, as part of the vision for genomics in the United Kingdom, as a Pathfinder project in which we are leading the world, the ambition to sequence 5 million genomes, which will be a mixture of whole genomes and other genomic tests, and that will be over the next 5-year period. So just when we all thought we might be able to put our feet up and have a bit of a rest, it looks like we've got another job to do. But the great thing is that through the National Genomic Medicine Service, there is, through the hard work of our ethics team with us members of my science team and NHS England, we're bringing live a single unified consent for all genomic testing in the NHS, and that means every single person who has a genomic test will be offered the opportunity to take part in research. And the research categories that allows is the ability for researchers here in the United Kingdom and across the world to work in our data centre on the data, for us to be able to work with commercial companies to undertake research and bring trial opportunities to patients. And then, very importantly, longitudinal life course follow-up of everybody who consents for research, and the ability to recall those participants for additional sampling or engagement in clinical trials or other studies as needed. And that therefore means that the concept that many of you are part of the Genomics England Clinical Interpretation Partnership, is now going to be not just 100,000 genomes, but an enormous research opportunity. So I'm going to stop there, because this evening is about hearing from people who are doing this and leading the research, but you now have the most fantastic opportunity. You're welcome later on to ask me or the team here who work very hard on your behalf with other members of the team of Genomics England who are here but also not here this evening, to try and create a research environment that's usable and effective for prosecution of research. And you'll see from Will, at the very least, the beginnings of some exciting data. And I'm expecting a lot of you, because it has not been easy to establish this resource, as you'll imagine. and to make it available in this way. And nobody's really done this in this kind of way with almost 3,000 people working on lots of different projects. The astrophysicists, when they do this, they usually work on a few discrete projects and they all work together. We're doing something that's very challenging, and therefore you will need to bear with us somewhat as we explore and discover the things we don't like mutually about our research environment, as Will has done and now has got through that phase and out the other side. So I'm going to introduce Will now and invite him to give his talk. So William Cross graduated from Cardiff University and he worked there in neurogenetics, and then went on to do a PhD in cancer genetics here at Barts and the Barts Cancer Institute, and then moved to work with Ian Tomlinson, who's sitting there as a postdoc in the Oxford Wellcome Trust Centre for Human Genetics, and then he came back home to Barts, and he's now here with Trevor Graham, who's also here in the front row. And so, without further ado, Will, it's my pleasure to invite you to present on the 100,000 Genomes Project, key results and lessons learned from the colorectal cancer domain. Thanks very much. There you go. I'm trying to give you my phone. That won't work.
Thank you.
All over to you. Thanks.
Can you hear me okay? Excellent. Well, thank you for that nice introduction, Mark, and really thank you to the team for organising this event. It's a bit of an honour to be one of the first speakers to come in to do this. So the overview of the presentation that I want to give today is thus. I'm just going to give a brief introduction to the GeCIP workforce, and this is just going to be very brief. But there's actually within that one sort of note that I'd like to get up front, which is 3 points why genome sequencing is so important. And I'll get to that in a minute. As well as that, I'd like to talk a little bit about how we started up with this project. So what we know about colon cancer, and importantly what we don't know, which leads really to the sorts of research questions that we generate when we go forward with this project. As well as this, I want to talk about some results, but this is a really important point that I need to make, which is that the results that I'm going to show you today are really just sort of skimming the surface of the things that are going on in the domain. They're sort of the key points that I thought were important to say today, but there's actually a whole raft of other things going on, and it's certainly not just me. It's at least 4 or 5 different labs that we have working with us doing this project, some of which are here today. So that's important to recognise that I'm but the sort of person who's here today. There's also a whole raft of other people doing work, very importantly. I'm just going to give one last slide as well, which will just be to describe what we've sort of learnt in general terms from doing this sort of project. It's quite complicated by its nature, so there is some lessons to be learnt there, and there's also some slightly more obvious things to say about it as well. So to begin, what's the motivation for sequencing cancers? Well, you know, the World Health Organization points this to be 1 in 6 of all deaths are associated with malignancy. And Bert Volgenstein has quoted many times that cancer is in essence a genetic disease. So it's to genes and it's to DNA that we must look for the answers. And really, the clinical benefit of all this is just starting to be seen, but there's much more that can be had. We've got this sort of idea of targeted therapies that Mark has already mentioned, and we've also got this idea of genetic subtypes and stratification. This is really a way that we can benefit patients just from genetic information that we know now, but there's a whole raft, I suspect, of information that could be beneficial in the future. So what is GeCIP? Well, GeCIP is this sort of workforce model, that's how I think of it, and it's the clinical interpretation partnership. So it's this idea that you can bring together different collaborations of people from all different backgrounds to work in one environment together to produce knowledge. And there's several points to this that are quoted on the GEL's main information website, but it's this one that I think is the most important. It's this idea that it could be sustainable, because there's a bit of a temptation with these sequencing projects to sort of do a lot of sequencing, produce a large paper, get it published, and then sort of go on and do something else. But I don't think that's what we're trying to do here. I think what we're trying to really do is set up a sort of actual framework, a place where you can come back and you can ask questions that come to you a lot later. Moreover, I mean, as Mark has already said, unfortunately, some of these people have very bad outcomes from their cancer, and we need to follow this up. This is not something that you can do just sort of one paper now and then forget about it. So this idea of sustainability, I think, is really important and underpins the whole idea of this GeCIP workforce model. So what is the colon cancer domain and what is it constructed of? Well, Professor Ian Tomlinson's at the helm, and we have 4 group leaders that work in coalition, and as well as that we have 15 postdocs and researchers and students, many of which are with us tonight. So this is really a team effort. And at this point I'd just like to sort of say about this question of, why bother with whole genomes? Exomes can do the job. I've heard this many, many times when people say this to me. Well, I'm going to give you 3 points why. Point number 1, exomes are only really sort of 1 to 2% of the whole genome. And I mean, it's an obvious thing to say, but if you're talking about targeted sequencing experiments, you're talking about a minor number compared to even 1%. There's a whole raft of evidence that suggests that non-coding parts of the genome are massively important for cancer. And actually, they're pretty understudied, generally speaking. We've really focused on things like driver mutations and coding sequences. But actually, the non-coding is the sort of, last stand saloon, really, of cancer, because there's probably lots of information there that we just don't know about. There's also significant analytical gains to be had from this, because when you have whole genome sequencing and you want to produce a model, or you want to do some sort of experiment bioinformatically, you've got an awful lot of data points to pick from when you look at whole genome sequencing. When you're talking about exome sequencing, as I've just said, it's a fraction of the information. Moreover, the definition of the exome is sort of changing, it seems to me, because when you look at older data sets that were exome sequenced, sort of 5 years ago, 10 years ago, there are genes that are not in those panels that we now know exist. And the same can be said for the non-coding parts of the genome. The definition of an exome is not really fixed, but if you sequence a whole genome, you're future-proofing the studies. And this is back to this idea of sustainable information. So those are 3 good reasons, I think, for why we want to do whole genome sequencing projects, exactly as GEL is choosing to do. So I'm now going to move on to talk about colon cancer and what we really know and what we don't know about colon cancer. So it's a bit of a complicated malignancy. It's certainly one of the biggest sources of death in the UK as well. Like I said, there's 40,000 cases a year and 1.8 million worldwide. And there's this sort of massive amount of complexity. It's a fairly sort of large organ, really, and it's geographically there's lots of information and lots of differences in the pathology of this disease. But there is something really important to say about colon cancer, which is that it's one of these cancers that we've made massive improvements in. And this is some statistics just from Cancer Research UK's website, where they say that 31% of bowel cancer mortality rates have dropped by 31%. This is substantial, and this has all been done through research. So we should really sort of look to this as motivation in what we're doing. And what about the pathology of colon cancer? Well, if you open any textbook, you're going to see something like this, which is the adenoma-carcinoma sequence that was developed by Basil Mawson. And this basically is this idea that you have this histological changes. So you have normal tissue here with these crypts of lubricant, and then you have, this is an en-face picture of a crypt focus. You then have an adenoma grow. These are not cancer, these are just the sort of preludes to cancer. You then get an invasive carcinoma developed. So that's pathology, but what's overlaid on the top of this is what most people refer to as the Vogelstein model. And this is really the often assumed truth. That's what I sort of tend to call it. It's an assumed truth, because if you open any textbook, this is what you would see. You will see a picture like this, and you'll see various genes. It's normally APC, KRAS, P53, and sort of others, put in this sequence to say, when you develop a crypt focus or an adenoma, you tend to have APC and KRAS mutated. When you go on to a serious disease, you tend to have P53 and other mutations. So the assumption that I think is there is that there's a sort of linear sequence, and that means that the more mutations you have, the more drivers you have, the more invasive your cancer is. Now, I'm not, of course, saying that Vogelstein explicitly says that, or that everybody explicitly says that. But unfortunately, when you open any textbook, this is sort of what it gets at. And there is this sort of underlying assumption from everybody that this is the way that cancers work. particularly carcinomas and colon cancer. So you have specific driver mutations that are strongly positively selected. This causes cancer. So this is sort of one of the things that I want to talk about today, and this is one of the many things that we were talking about when we started doing this study. So as well as the pathology, we've got sort of two main subtypes that are often looked at with the genetics of colon cancer, and that's MSI status, which is this microsatellite instability. And that's this idea that you can have a hypermutation rate. The alternative to that is MSS, which is this idea that you have normally a high level or moderate level of aneuploidy, and you have Wnt disruptions that drive it, which would be normally the APC mutation if you're looking at the Volgestein model. So what is aneuploidy? Well, aneuploidy really is just an abnormal chromosome constitution. So it's this idea that when cells divide, you can have balance changes in the number of chromosomes that they have, and this itself can act like a mutational process for cancer. So I'll talk a little bit about that later. And as I said, there are many driver mutations that are known in this process, and they do relate to these subtypes in many ways. So just very, very briefly, what do we mean by a driver mutation here? Well, we're talking about a mutation that is acquired within a gene that then gives the cell a phenotype for malignancy. So that's, this term driver is bounded around a lot in the literature, but basically it's just something that is sort of associated or in some way causal with the pathology of malignancies. So that's just a definition now. Just thought I'd put that up for clarity. What other considerations did we have with this study? Well, one very clear picture that was beginning to emerge when I started my PhD in 2013 was that you've got an enormous amount of heterogeneity, both inter and intra-tumour heterogeneity. So what we mean by this is that if you profile these tumours within people, you actually don't obviously see the same mutations. You see different variations of mutations, and most of the time you see different types of genes that are mutated. So it's this idea that you've got diversity, really. That's all it really comes down to. So, I mean, how does this fit with the classical model? How does this fit with this idea that when you open a textbook and you've got a fixed number of genes, but then you've got all these sort of papers, including some of our own that we just published earlier this year, and also the TCGA efforts from a few years ago that say you've got this huge amount of heterogeneity. So there's a bit of a conflict here, and this was something we were thinking about when we embarked on this study. So I suppose really if you want to pitch this as questions, it's sort of, do all colon cancers have these similar sort of disruptions, even if they're not the same mutations? Are they the same pathways that are disrupted, or are there a core set of pathways that can be mutated? And what about these sort of newly identified macromutations? Because there are lots of things like chromathripsis, genome doubling, lots of other mutational events that are without doubt important to the pathogenesis of colon cancer. But actually they're not very well explained, or they're not very well even explored at the moment. So these are the sort of main things that we went into this study with in our heads. This is just the brief outline of the workflow. So, I mean, as I said at the beginning, there's a whole lot of people that are involved in this study, not least the participants and the primary care centres that take the samples. We've then got the Genomics and Bioinformatics core that process this data up front, which is what I would term pre-processing, and they obviously produce their clinical reports on this. And then this line is supposed to indicate the sort of research environment, which is the things that we would do as researchers and the things that we might want to do that are not necessarily already known about. So as we go on to talk about some data now, the two things that I want to focus on really are this idea of driving mutations, and I'm going to touch a tiny bit upon this idea of aneuploidy as well and what that might mean. Just some summary statistics here. So we've got, at the moment, just over 1,500 participants, but there are obviously more because we've just had a data release, in fact, about 4 hours ago. That's the number of tumour normal pairs that I pulled out just a few hours ago when I looked on LabKey. So that's a substantial number of patients that we can now interrogate. The clinical information we've also got, it's somewhat incomplete at the moment and it's ongoing, but we've certainly got information for more than 700 participants in terms of TNM staging and other pathological information that would be important to interrogate. And this is the first of what I would call scary diagram. It's scary because there's a lot going on, and I think if people aren't perhaps used to looking at these graphs, they think, what the hell is going on here? So we'll just go through it bit at a time. It's very difficult to try and sort of get these figures together where you've got over 1,000 patients worth of information and you're trying to put it all together. But this has been done by the TCGA, so I can't claim sort of, benefit for, I can't claim that I invented this, but essentially you've got on the columns one patient, so each of these nicks represents a patient and the mutations that this patient has. So this bar chart, this scatter plot at the top represents the number of total mutations across the genome that we've recorded in this patient. And as you can see, if you scan across, each of these black dots represents the total. So that each one of those is a patient. And there's a huge variety. That's actually a log scale, just so that I can fit it all together. There's a huge variety in the amount of mutations that you see in these patients. And the first thing you notice on this is that MSI obviously drops out straight away because we know they're hypermutated, so they have orders of magnitude more mutations. But already you can see there's something a bit interesting here because they've got loads of indels. They've got nearly as many indels as they have total. So in other words, the number of SNVs is not as many as you would think, considering they're microsatellite unstable. The opposite is true when you're talking about MSS cases. They have less indels than they do SNVs, point mutations. That's quite interesting. I didn't necessarily expect to see that. These are POLE and POL-D cases. So those are a very rare subtype that you see in colon cancer, and these are so-called ultra-mutated cases. So these are sort of less than 1% of colon cancers would have this particular phenotype and pair of genes mutated. What's nice about having over 1,000 patients, although it's not nice for the patients, but it's nice in terms of the science, is that you can actually pick up these rare cases and have an opportunity to study and learn from them. I don't know how many POLE cases have been sequenced around the world, whole genome. I would imagine not very many at all, so this is a very unique opportunity. One thing I wanted to point to here as well is that the reason I've coloured these mutations here and these genes, so this is, for example, APC, which I mentioned earlier, which is part of the Volgenstein model, and then a couple of other genes that are commonly seen as part of that process as well. I've coloured them because you can sort of almost, without even doing any statistics, see what's going on here. You've got near ubiquity of APC. Less so in the MSI cases, but near ubiquity. BRAF is heavily enriched in the MSI cases, and we know that from lots of studies already, but it's certainly nice to see it in this data. P53 and KRAS are enriched in the MSI cases. But here's the thing, there's mutual exclusivity between BRAF and KRAS. You can see when the patient has a mark here, they don't have a mark in the purple, and I think there's only 1 or 2 patients that actually have both. So why would we care about that? Well, this sort of thing is really important because the RAS pathway is a drug target. It's currently a drug target, and there may be potential in there being more targets in the future. So how these mutations are distributed across these different subtypes that we might find, very important. So moving on, we can sort of look beyond just the few mutations that we tend to focus on, and we can look to the frequency of other driver mutations. So this is just the frequency of driver mutations that were identified using the MUTZIG algorithm, and this is the top 50 for both MSS and MSI cases. So I've split them off because there are different subtypes, as I discussed earlier. And we see our favourite genes at the top. We see APC, KRAS, P53, and we also see them to an extent in the MSI cases. We can also break this down by type of mutation. So in other words, we tend to see stop-gain mutations in tumour suppressor genes, and we tend to see non-synonymous mutations in oncogenes. And this is all sort of, we know about this already. But here's the thing, there may be lots of potential to try and understand genes that we don't know whether they're tumour suppressors or oncogenes just by their mutational patterns. Unless you've got 1,000 patients to pull this information from, you're not actually going to have the frequencies available to do it. So there's an opportunity to actually learn lots from this information. And as I say, this is ongoing work. If we just compare by we just take the top 5 and we just say, okay, well, what's been found before? TCGA found that APC, P53, KRAS, a gene called titin, which I think is very much in question at the moment because it's sort of a large gene, so it may not be actually to do with cancer at all. PIK3CA and this FBXW7. We didn't find that. We found mostly the same, but we found that TCF was more common than FBXW7. So it's a little bit different. different enough that I should think you should take note of it. If we look at MSI cases, we really do see differences there, because their cohort was only 30, our cohort is sort of nearly 200. So the power of the information to say that BRAF is actually the second most common mutation, and this ubiquitinase gene is also the first. I mean, this is very significant, and I couldn't explain to you why, and you know, this is something we need to investigate, I think, further. But what do these frequencies actually mean? Because we can sort of put all the patient information together, and we can say, well, by frequency we see this gene and we see that gene. But actually it's the individual patients that sort of we want to glean information from. So what I'm going to try and show you next is some of the analysis that we're trying to do, which is to try and deconvolute this information, to try and understand combinations. So I'm going to have to show you another scary diagram, I'm afraid. Do the frequencies really represent anything in individual people? Well, no, of course they don't, because you're losing that information by merging them together. So that's the motivation for trying to look at mutational combinations. And this is another scary diagram. Each patient is on a column here, and what I've done is used hierarchical clustering to group the patients by the types of mutations that they have. So it's a sort of ordering method. I think you could very simply just think of it as that. And the colours are based on the groupings themselves. So straight away you can spot the MSI cases that naturally fall together within this clustering method because they've got lots of mutations. You could even just see that by eye. Now there are lots of other clusters here that you can identify. And the thing is, if you just sort of pick out a few of these that have unique mutational combinations, and then you look at the frequencies of the genes within the clusters, you start to see a completely different picture to what you would have seen if you would just put all the genes together, as I did on the last diagram. So for example, this is the MSI cases that you've seen on the other slide. This is another group that just exclusively have ARID1A in 100%. That's 24 patients. This is another group that has FBXW7 with APC. Again, 33 patients, a significant number of people. These combinations of mutations are not in textbooks. This is not sort of, you know, colon cancer has this gene and this gene. This is sort of getting to the nitty-gritty of what individual people have in their genomes. I'm just showing you this. This is a very sort of, you know, in a way it's quite a cursory piece of analysis. I think there are much better ways to do clustering, and we're certainly going to do them. But this does demonstrate a point, which is that as we produce the first publication from this piece of work, we need to discuss and make a point of the individuality and the heterogeneity of these patients. So there are a massive number of driving mutations, which I think is obvious, but there are actually a massive number of combinations. I think that's what I'm trying to get at here. And even if we just restrict, if we just said, okay, well, let's just restrict everything and we'll just look at APC, KRAS, P53. You know, those are the three genes that I've mentioned it several times already. It's in every textbook. Let's look at the combinations. Well, you actually only see the golden three, APC, KRAS, P53, in 39 patients. You don't see it in anywhere near high frequency as you would expect to see. Even if you look at other KRAS types of genes, you don't see a significant number. And actually 98 patients just have APC on its own without even having KRAS and P53. The most common combination, if you were to just look at these three, are actually APC P53. So if you're going to argue that there's any sort of relationship between two specific genes and colon cancer, it's without doubt those two, but KRAS I would say not necessarily. Just for the last part of this talk, I'm going to talk about aneuploidy, and this is because aneuploidy is a very important and significant part of all carcinomas. 80% of carcinomas have a degree of aneuploidy, but really it's anyone's guess what it means. It's very much debated about the processes that underpin aneuploidy, and it's very much debated what it means in terms of pathogenesis. In other words, we don't actually know whether copy number changes act as drivers. The assumption is that they are, but this is all sort of information that we just don't know. So just to explain what I mean by aneuploidy, when a cell divides, you get these chromosomes neatly forming into partitions in the two new daughter cells. When you get aneuploidy, this process breaks down. So you end up with chromosomes being gained and lost in the daughter cells. And this broadly, you could just describe this by the term ploidy. That's what lots of people would say. You start with a diploid cell, or a two-end cell, then you end up sometimes with genome doubling, and you can end up with this sort of fragmentation events where the chromosomes start to break apart. So you end up with a totally sort of messed up genome, for a better word. And incidentally, this figure here was taken from Ian's review from a few years ago that he wrote. Very nice review, I'd recommend it. And so we decided to do a piece of analysis using a tool called Sequenza, and this is with 2 other members of the ICR and our lab here at Barts, which is Jacob and Julio, and we developed this workflow where we can process over 1,000 patients and we can QC each patient, because one thing about dealing with 1,000 patients is that you can't just blindly run tools and then sort of take the results as is. You actually need a lot of QC in place to make sensible results from them. And it's a fairly substantial piece of work just to produce this pipeline in itself. So this is what we've been doing, and I'll show you the results of this now. So this is what it looks like when you take 1,000 or more genomes and you plot the gains and losses. So another scary diagram, I'm afraid. This is chromosomes 1 to 22 across the bottom, and this is sort of the genomic position, just ordered 0 to 3 billion. This is, I've marked the information that you've already seen. So these are the MSI cases, these are genome doubled cases. So the thing that I find massively surprising about this is the number of genome doubled cases. We've known about this for a while. I think for decades people have been saying aneuploidy is associated with genome doubling and there's some sort of relationship here, but that's nearly half of MSS cases have this genome doubling. So this is clearly a very important mutational process in colon cancer. Yet if you look at the TCGA's paper, it's not even apparently in their vocabulary. It's not even in the paper. So I'm just going to show you a frequency plot because this is sort of an easier way to interpret what's going on. These events marked in red are gains, so this would be where you've got more than 2 chromosomes. And if you just look at the gains, you can actually start to see a trend where in MSS cases, this middle block, you tend to see gains in 7, 8, 13 and 20. And again, some of this we've already known, but there's an awful lot of detail if you look here. For example, that 5P arm seems to be gained in a lot of cancers. This is new information. We don't know what a lot of these things mean. So I'm just showing you this because this is sort of how many questions can you generate from one plot? It's quite a lot. And it really remains unknown at the moment what this means clinically, but I think in time we're going to have the information to actually try and understand this. It may very well be that genome doubling has clinical benefit. Maybe it doesn't, but there's lots of biology under this as well. I mean, we do need to remember that as well as trying to produce information that's useful in the clinic, there's actually a whole raft of biology that's useful. So this is just one result that I'm going to just share with you now that I think will probably end up in our first publication, which is that if you take the P53 status of these patients, this is just grouped by poly-MSI genome doubling MSS, and you say on the left there, those bars are P53 mutant patients, those are P53 wild type. The assumption would be when you have a P53 mutation, you have lots of ploidy, you have lots of genome fragmentation. That's sort of, again, if you open lots of textbook and you read lots of papers, that's the sort of things that they say. Doesn't look like that's the case at all. In fact, the only significant result is that when you have genome doubling, you have obviously more ploidy. When you have genome doubling, you have a higher propensity to have fragmentation. But within the groups, P53 doesn't seem to make any difference. Again, that's massively surprising. But this is a large number of patients. This is 1,000 patients in these plots. So this has to be a significant finding. I don't know what people are going to say when we say that P53 isn't as important as we think it is, but that's what the results say, that's what the data says. So I'm just going to give you a very quick summary now, and then that will be the end of the talk. So the summary of findings really is that the landscape of colon cancer is very much complicated and varied. It's an obvious thing to say, I know, but I think it's a lot more complicated than we gave it benefit for. There is this convergence on Wnt, RAS and P53 pathways, but there's a whole lot of other pathways there. Those genes that I showed you earlier, that's just the top 50. The tails go on for a long, long way. The Volkswagen model doesn't really fit the data, and I mean, I know it's not to be taken implicitly anyway, but we really need to move on from this idea of there being sort of 3 genes that you can describe cancer by. And there's potential to build much more robust and much more beneficial genetic classifications using things like genome doubling and the degree of shattering. As I said at the start of this talk, I just wanted to point out that there's a whole raft of other things going on. This is just, you know, a few results that I've put together into a 20-minute presentation for you that I've personally been doing. But there's a whole lot of other work that's been doing, and this is one of the slides that one of our other members of the group put together, which just described some of the mutational signature profiles that he's been looking at. So again, a very complicated piece of analysis. So what have we learned? This is just the last slide, and I'm going to try and sort of probably preach to the choir, I imagine. But teamwork and communication in a project like this, where you're dealing with thousands of pieces of information, thousands of BAM files, FASTQ files, you have to have an open communication channel. You can't run the group with individual people trying to take over and do pieces of analysis on their own. It won't work. You have to have regular video conferences, task delegation, but you also have to have freedom. You know, everybody should be sharing data, and I'm sure most people are. You need to have a situation where the data is there, freely available for everybody to use in the sustainable environment that we spoke about, so that you can come up with ideas and you can explore the data and find out new things. Another sort of thing that I just like to say for the bioinformaticians out there is that, and I know most people would just do this anyway, but you need to write efficient code, and you need to write code that can be reusable. There's no point in sort of writing a script that you can just run once just about and then it breaks the next data release. It needs to be, we need to write code that anybody can come into the environment and use. And the Genomics England bioinformatics team, really they're an amazing resource, and there's several people that I've been bugging all the time on email saying, I'm sorry, you know, so I have to ask this again. They're very helpful, and constructive criticism is generally very well taken, I would say. The project is obviously far broader than any individual domains, and I think going forward there needs to be more crosstalk between the different cancer domains themselves. So it's just an acknowledgement slide, and Sarah's been very helpful over the last few months, organising meetings and also putting up with me asking strange questions on emails. Narupa as well for all the hard work collecting some of the clinical information that we've had, and Chris Odhams as well, because I know particularly of all the other bioinformaticians, I've emailed him a lot asking different things. Can he install this tool? Can he install that tool? Can he change this? So he's put up with a lot, I would say. And that's all I have to say. Thank you very much.
Thank you very much, Will. That was very encouraging. So it's now my pleasure, this will be the format for how we do these moving forward, but it's a real pleasure to welcome our next guest, who's Malak Abed AlThagafi, who is a well-renowned Saudi clinician scientist who has spent 11 years in the United States at august premises such as the Dana-Farber Cancer Center, UCSC, and Georgetown, and is now back in Saudi, a leader of the Saudi Human Genome Project. And it's a real privilege to welcome you here this evening to speak to us and share with us the wonderful work you've been doing there. So Malak, it's over to you. Thank you very much. Will, can I just borrow your pointer? Just in case you need it, that first button.
Well, good evening, everyone. Can you hear me very well, or should I move the... Okay. Thank you for having me today. I'm honored to be here, and thank you for the kind introduction. So I wear multiple hats, as you can see, as a physician, as a researcher. So my clinical work, I do it in the molecular diagnostic, my research with the Saudi Human Genome. Now, I want to share with you the part that maybe not many of you know about it. So my last visit to London was in 1988, when I was around 5-year-old, so now everybody knows my age. And this is my pictures, actually, this one, actually in the hospitals here in UK, in London. I asked my mom, do you remember which hospital? She said, I don't know, maybe it has some king's name on it, so I assume it's King's Hospital. So that's what she said. So I was born with the genetic disorders, and at that time, in the late 80s in Saudi Arabia, you can imagine, get diagnosis and treatment, and to find a genetics a doctor who specialized in genetics actually was very difficult. There's only one physician at that time who established this clinic to serve the entire kingdom. So I had to come to the UK, spend 1 year, and this is the surgeon who actually treated me here. And from that, I started to love and have passion toward medicine and genomics. So as you heard, I spent almost a decade of my life in the US, then I decided to return back to Saudi Arabia almost 3 years ago. And I wanted to work on the area I specialize in. I specialize in molecular genetics along with other fields in pathology. But I found very nice alternative niche. And that was, I came in the right time to find out that the government decided to establish a national program. It was announced in early January 2014, and they called it the Saudi Human Genome Program or Project. So like any national project, they wanted to start it with 100,000 subjects. And the focus was in the beginning on discovering a variant on gene that is responsible for specific genetic disorders. So it was looking at the monogenic approach to different genetic disorders. And this is, of course, important. They wanted to implement and provide infrastructure to be able to do this kind of testing. It's funded by CACST, where it's King Abdullah City of Science and Technology. You know, this is not started in Saudi Arabia. There are multiple projects along, national projects, for example, the Veteran Genome Project in the US, that looks specifically in the profiling of the veterans. Some other, as we heard, an excellent topic before in UK, they're focusing mainly in cancer and some rare genetic disorders. And here in Saudi, we wanted to focus on specific genetic variant. This is not only unique to us, there is multiple other efforts in the world, and actually I cannot update, I didn't update the slide, but I think there are even more new projects in Japan, in China, everywhere in the world. What's unique about this part of the world is how the highest level of consanguinity, or the habit of marrying first cousin. And it actually is true. In Saudi Arabia, in some region, we have this can get up to the 80% of, in some area in Saudi Arabia, 80% of marriages happen between first cousin. On average, actually between 40 to 60%, which makes this population is very interesting population. It's like walking in genomic lab, just studying people. So people already knocked out, so you don't have to knock out anybody. So this is how it started. So today I'm just going to give you a flavor of everything going on. Unfortunately, 99% of what you're going to hear today is not my work. It's been done by other people, but I'm just honored to present them today. So the central lab is in Riyadh, in CACST, here in the middle, and this is the CACST. And then the idea is to have different satellite sites around the kingdom. Currently, we have 7 sites covering covering the east, the west, the south, and a little bit north and north. Why we wanted to do this? Of course, government, when they want to fund any national program, they wanted to look into 2 things. How this will improve the economic burden on them, and this is one is the idea. Maybe by understanding the genomic, we can lower the healthcare costs on the kingdom, on the government, and also they want to improve the quality of life. So the proposal was, by identifying this risk factor or diseases that are unique in Saudi, that will help you to establish a better screening program and effort, preventive efforts in these populations. And also it enable you to implement what we know as a personalized or precision medicine, which means you can write for the future generation, maybe you can design a better therapy especially for these rare disorders. And also, socioeconomic impact. You know how the world is growing, especially in the field of genomic industry. And I'm going to show some numbers later on just to tell you. So we wanted to enter to this field. We wanted to able to transfer the technology, to train the locals, and also to increase the jobs in this field. With that, we thought whatever, with the kind of population and homogeneous population and data we can generate, we actually can be leader in this field. And there is a say, which my boss liked to say, our next oil is actually the genomic data we generate. So that's what they wanted to do. Actually, the benefits go beyond this. We go beyond the human. We think whatever we build will help us better in developing different programs relating to the humans, and infectious disease, and food safety in animals and planets, in biofuels, etc. So the goal is to come up with this national genetic ID, hopefully for every single Saudi citizen. So as I told you, so how does this project work? It's a national program, as I said. CACST has the central facility for it. However, we have different satellite sites and different institutions. And each institution has its ability to get their own IRPs approved that been reviewed by a central committee. And then the satellite site has its choice and freedom to either work on separate problem or even to work with others. And with that, we also try to control and unify the protocol, sample processing, QC, and QA, as it was mentioned. It was one of the very difficult things to do when you work in the national scale. So you have to really to put some standards so you get some meaningful result. So the phase one of the projects started early in 2014, and basically it was a phase where you wanted to bring this technology to the kingdom. So you wanted to establish the lab with the infrastructure, work with different satellite sites, trying to see which one actually can deliver. So we started actually with 14 sites, and then we dropped it to 7 because some sites, they were not really up to the level that we were looking for. With that, we started very small, so we're a baby project compared to the UK Genome Project. So we used Thermo Fisher Technology 25 sequencer, and this is the data side we generated every year. Now with phase 2, we're getting a little bit bigger, so now we're introducing the other platform to start to do a whole genome sequencing. So in opposite to what we hear, the UK Genome is actually a whole genome project. The Saudi Genome Project is not a whole genome project when it started. We started with different techniques. And this is now the data size we can generate every year. And this is what we do. So we use different modalities for one sample. So we have biobank sample from different rare disorders and families that we collected over the years. And we tried to work with actually certain panels. We try to learn from to start from what people come up with. So created some panels at the beginning. And sometimes the panels work, so you detect a reason, a cause of it, and sometimes not. So when it doesn't work, we go to the exome. And with the exome, we've actually been very successful in terms of utility for diagnostic purposes and even for discovery. So you heard in the beginning, the detection rate here was around 20 to 40%, if I'm not mistaken, depending on the disease. In Saudi, actually got to 60 to 70%. So, and later on, we're moving this rare diseases to the whole genome. And the other reason why we have whole genome now, because we wanted to start to do cancer as the next phase of the project. So the following few slides, I got it from our bioinformatics team, thanks to them, because really without their work, I don't think this project would be going anywhere. So they've really been doing a lot trying to help us with this project. So this is what we have. So CACs have this ability of generating data with a fast clouding system from other satellite sites. Everything gets centralized in this data center in CACs, where it has this 5,000 CPUs and ability to store it at 15 pp data size. So that's what we have up to the data I got up to August of 2018. So we had almost 43,000 run from 37,000 patients and families, and 42 different study projects focusing on monogenic disorders as phase 1, and 35 different centers, and has more than 90 plus primary investigators and over 120 users. And that was the variant result we have so far till August. 4.3 million variants that can call, 7,000 of them actually common in Saudis. And this information, we wanted to use it immediately for our diagnosis, at least for some of these rare disorders. Before starting this, I just wanted to share with you some of the computational challenges. Maybe most of you who work in this field know about it. So Saudi Genome Project, like any other big genome project, is a big data problem with this famous 4 V's, the volume, veracity, variety, and velocity. While we have another issue by having this different location, which some of them are really far away from Riyadh, so it's really hard to get all people to work on the same term. So you have here big data, and you also have complex level of analysis. Adding to it is the other issue, privacy and cost and management and security. So you really have, so establishing this high-performance clouding system to try to work in parallel algorithm engineering and workflow system become one of the top challenges our bioinformatics team work with it. And this is just information, which is funny information. You know, sometimes we don't imagine how much data we generate. But a project, a famous project like the 1000 Genome Project, when it started, generated in the first 6 months, what entire gene bank produced in 21 years. So imagine what UK Genome Project produced, what we produce, what the rest of the world produce. So there is a big the data problem, if you will. So this is here just to show you different level of analysis we do here. We just put a tumor, even though we just started in tumor, but it's actually applied for different platforms. So, and sometimes we enter where we go between, as I mentioned, between different modalities, and then there are different level of variation, annotation, and calling up to the level when you get the report. Primary analysis, secondary analysis, tertiary analysis, also that to make it make sense with the phenotype we see and the clinical information we have. So this is what our team just we've been doing as internal bioinformatics team. So they have created a total of annotation tracks, over 100 tracks. And they do it really depend on different kind of level or layers of information if you want to think about it: positional, functional, population, clinical, mutation effect, and knowledge-related variants. Oh, sorry, I think I'm going back. Okay. So why we need this? Because with this kind of information, in a country where burden of consanguinity and very high carrier level, which wasn't very accurate, at least before we started such a project, we did not know the estimate of autosomal recessive disorder diseases burden in Saudi Arabia at this scale. So now we can have some of the probability number that can be used later for better screening programs, especially when it comes to premarital or newborn screening, etc. So now we know we have at least one founder mutation, 1 in 5 from our population. If you're looking for 2 founder mutation, 2 in 100, at least 3, it's 2 in 1,000. And this is the top 3 disease group which we have it. The psychomotor disorder, 3 in 1,000, retinal dystrophies, and congenital glaucomas. So there is over 100, I think, publication up to date related to the Saudi Human Genome Project in the field of monogenic disorders. And here I want just to highlight some of the key publication and why how it's helped us at the national level. So here, for example, we showed actually the panel sometimes can work better for us than doing the clinical exome in terms when you want to report to the patient in real time. So here we show actually you can do a little bit better by using this panel as an initial step when it comes to translation, if you want to translate this to the real world. With that, we also we discovered around 440 what we call them non-Saudi mutation. That means they're very common in our populations. The second one is talking about, again, finding this, the carrier disease mutations, which is now the last update I have, it's 1 and 2, actually. If you have 2 Saudis, one of them carry some sort of mutations. And then the third thing is actually to use this database and trying to reclassify some of the disease variant in the public database. In this paper, there was a reclassification of around 2,000 variants from ClinVar and HGMD, for example. In addition, you know, when you do this kind of studies, especially in homogeneous population, you discover of a lot of gene for unknown phenotypes. So this was the theme for it. Now, there is one drawback I have to tell you, and I think it's true even with other national projects. We are at the very fast rate of kind of making this correlation genotype, phenotype correlation. However, we still have to catch up in terms of doing the functional work, the biology work, to see the real causation, if you want. With that, I just wanted to show you here the impact of doing such work, how it actually changed your diagnostics. So from here, in this paper, from the lab I work at now, the diagnostic clinical lab, where we looked at the 1,000 first diagnostic report for panels or whole exome. And our detection rate for finding a meaningful result were around 60 to 70%, which is higher than what's been reported in other programs. And this is supported diagnosis actually in around 5,000 cases, individuals and families. And also with this knowledge, we now been able to work more in the prevention genomics, if you want. And I want to talk more at the end of my presentation about this new program of expanded premarital screening in Saudi Arabia. So this is what the clinical diagnostic lab benefits from such work at national level. So how you translate this discovery to meaningful information to your patient at the right time. As you heard, most of these diseases are rare, and sometimes of them are they really don't live for a long time, unfortunately. So maybe we cannot help these patients because we don't have the current therapy to help them, but probably we'll have to prevent the family from having another child with a similar condition, at least when you give them an answer. And this is what the lab did. So collectively, we have different panels in addition to doing the clinical exam for 37,000 genes that cover around 500 hereditary disorders. And this is one of the most comprehensive panels in the screening panel. It's very unique for the Saudi population and Arab population in general. And up to date, I get this info that September 30th, we served around 30,000 samples working on these panels and clinical whole exome testing. And this is very powerful for a population because it's really plotting it's not only against the known international database like ClinVar or GMD, but also towards the Saudi Human Genome database, which has over 40,000 subjects. So I told you we'll hear a little bit from everything, so bear with me. So now we're just going to switch topic a little bit to cancer, which wasn't one of the more focus of the Saudi Human Genome in phase 1 at least. But I just want you to read this statement, which I took it from the WHO, one of the recent WHO publication, which is say, the Gulf country states actually are forecasting to have a sharp 150 to 200% increase in cancer rate by 2030. This is one of the highest expected increase in the world. And we don't know why that. It could be, cancer is a complex, it's a genetic disorder, it's a very complex disease to pinpoint it to one cause. And this is the trends of cancer in Saudi Arabia. So that's the cost, what it costs the government every year. The gross rate is around 5% increase for this number of cases every year. We have every year around 20,000 newly diagnosed cancer patients. This is a total living patient. And this is a top, for example, here, number 2 in males, non-Hodgkin lymphoma. I don't think this is in a top 3 in any other countries I know, at least in the Western world. Usually colon, lung, prostate, or this in different orders. But here we have leukemias and lymphomas and liver are common here. Okay, we can blame the liver, maybe it's related to hepatitis or something else, but we actually have some de novo and family trend in some families, so we still don't know why. And in females, thyroid comes as number 2. And again, we don't know why that. And here is some number. I told you this is a very growing market, and this is the data you hear specifically for cancer and why you need to be in this next NGS era of cancer diagnostic or treatment. Cancer prognosis, global market level, $6.3 billion by 2020. Cancer monitoring market estimated to reach $10 billion by 2020. Cancer diagnostic, which means you use this technology for therapeutic and diagnostic purposes, is approximately estimated to be $31 billion. And this is in high and medium income countries. And this is by G. Morgan. So this is a very significant amount if you look at it by an economic, if you look at it from economic burden in different countries and life and healthcare systems. So with cancer, even that we didn't start with the discovery part and doing the whole genome part of it, we wanted to implement this at least at a diagnostic level. And that's what we started. So we wanted to make it available Unfortunately, we don't have the luxury of having biobank samples like what we had in our rare diseases. So here you have to establish a system to start, so you can do this whole genome discovery. But we started with a panel, and we wanted to do this at least to help patient to get the to help the patient to get this targeted therapy. And sometimes, you know, you want to know some certain genes, like for example in colon cancer, you want to know the patient has KRAS, so you don't then give them BRAF inhibitor. It actually will make it worse for them. So sometimes you want to know certain genotypes just to not to give some targeted therapy. So we started this targeted sequencing panels for cancers. It's kind of very standard panels. We used some of been available in the market, and then we created our own customized panel, depending on what information we found. So the report covered just a simple report to tell the oncologist what mutation type and some information about this gene or mutation, and then what trials or actionable drugs that they can use. Remember, you're trying to make it very easy for somebody who actually did not really study in depth genomics, and they're very busy in the clinic seeing 40 or 50 patients in one clinic. So you want to make it as user-friendly as possible to them. So this is what we did. This is the kind of the report. So what is the panel? You tell them there is one alteration, there is one drugs. That's it. And then you tell them a little bit what this drugs, and then you tell them this is the medications. So this is the kind of the simple approach we took for them. Now it will be very complicated with the more and more drugs and clinical trials being in the market. So it will be. We try with this also to educate more and more the new generation of starting from medical student to medical doctor in training, and etc. They have to deal with this information. Either they like it or they don't like it in the in the current or in the near future. Now in cancer, there is another exciting thing. Remember I told you for some reason there is estimation to have this sharp increase in cancer patient in the Gulf state for some reason. Some observational study and some actually small studies like most wanted to see why, for example, why Saudi women get breast cancer at least a decade earlier or younger than what the Western population. It's not unusual to see somebody who come to the breast oncology clinic with a breast cancer at the age of 40, 30, 20. Actually, last week, I had a case came to us. The girl is 10-year-old and she has ovarian cancer. So there is something going on, and we think it could be related to some sort of familial or inherited cancer that maybe what's known at international scale 1 to 5% that inherited cancer, maybe it actually has higher prevalence in Saudi. And some earlier study tell us it could go actually to the rate of 20 to 25% of all newly diagnosed cancer patients. They could have carry some of this sort of inherited cancer mutation. So this when it comes to germline testing for these tumors. So we established this small study. We called it the Saudi Familial Cancer Project. We wanted to recruit people on prospective way. We try to be as non-invasive as possible. So we started with saliva actually. Because remember here, if you find the cause or a mutation, you have to test the rest of the family. So it's really you have to make it as easy as possible to them to get enrolled. So with that project, it came with to establish the first cancer genetics and prevention clinic in the kingdom with a proper pre and post counseling for this. This is something maybe came late to the country like Saudi Arabia, but it's better to start now, not to start any time. So we started this just as 2 years ago. One lesson. Actually, it's very easy. If you ask people, people will tell you more things. So sometimes you just forget to ask people, especially in cancer clinic. Just asking them if you have anybody in your family, or sometimes people they just relate if somebody has other type of cancer in their family, maybe it's not related to me. So just trying to ask them and try to make them make their own homework. And remember, you deal with people with different backgrounds, different educational backgrounds, different tribal backgrounds. So for some of them it's kind of taboo to talk about it actually, and even ask their relatives. So you really have to tell them, we're not here just to know, and we try to help you and help your other relatives. If you know this early, you don't have to come to us at stage 3 and 4. we can put you in a special program so it will help you. Again, we do this because this is the goal of it, to implement a national surveillance program for familial cancer program, to make this technology very affordable and user-friendly. And also, we wanted to have the multidisciplinary team work on it, including not only the oncologist or medical doctor, but also the health policy expert, so they can adopt such policies, bioethicist, informatician, etc. And with that, we think we can create a paradigm in precision predictive cancer genetics and medicine prevention, true prevention of cancer in this special group of cancer patients. And here one example. We're still recruiting patients, but we've been getting a lot of segues to this project. This is a typical Saudi family. The parents are first cousin. They lost their son at age when he was 8-year-old from a rare type of lymphoma. And then they their daughter developed a nasty brain tumors called glioblastoma. For those of you who maybe you heard about it when John McCain and then Ted Kennedy before got it. It's a very nasty tumor, and usually in older people, but she got it at the age of 4. Then, of course, they took the standard care of therapy. So they removed everything surgically. They started to give her chemo and radiation. Unfortunate girl, with the moment they wanted to start the chemo after the radiation, she actually relapsed, which means like nobody touched the cancer. Everything came back. She relapsed on treatment. With that, the family refused to do any more surgery, and I don't honestly blame them, because she did not really benefit from that. So when we went back and asked them more, we learned about that son who was 8-year-old and died, and now is a girl, and there is some random cancer death in the dad side. So we offered them to do this testing, and indeed we found this one of the rarest cancer syndrome called constitutional mismatch repair gene. It's actually one of the few cancer syndromes that been transmitted in autosomal recessive models, which is so. You can see where I'm coming. It's their first-degree relative, so they could have higher probability. So, and then we wanted to take it a step further. So we told the family, there is a treatment. It's not FDA approved for kids to start with. It's not FDA approved for this for constitutional mismatch. But we see some scientific evidence in different types of cancer mismatch. MSI, for example, they get this kind of immunotherapy, or mismatch in general. So we told them, see, she has mutation of germline mutation in one of the gene. We can try to give her this immunotherapy. They said, you know, we have nothing to lose. Let's try. So it was a very difficult decision for them and for us, because if everything went right, everybody will be happy. If everything went wrong, nobody will be happy, and we'll be the one who blamed to use some kind of novel therapy. We did, and this is her true tumor before starting the immunotherapy. This is after 6 doses, almost shrank by half to 50%, and this is after 24 weeks. Today, the girl is alive, 17 months, with no relapse so far, and she started to go to school, and her latest MRI, I don't have it here, it actually a tumor, even I think what remained is less than 10%. So it's really working for them. This has opened the door to have a local clinical trial for this kind of patient with this rare syndrome. And what we found from at least initial recruitment, this pediatric patient with high-grade gliomas, at least 20% of them in our population carry mutation for constitutional mismatch repair. Finally, I will go back to another screening programs. I talked to you about it in the beginning. So with a country who has this huge number of genetic disorders, and also the habit of getting first cousin marriages, second cousin marriages. So you really have to think again, after all this knowledge, what kind of screening program do you want to use in your premarital programs? So our current premarital screening programs only cover 2 genetic disorders, thalassemia and sickle cell anemia. So, and I told you, we at least discovered there is 440 mutations that are unique to Saudi, and they are very common, 1 in 2. So if you're not including this kind of thing in your premarital, you really will continue the same close cycle of getting more and more genetic disorders. So the couple don't have they don't have any opportunity to know if they carry this mutation, if they want to go with this marriage or not. So now, working together with the Ministry of Health in Saudi Arabia, where they are the largest clinic health provider in the kingdom, they cover almost 70% of the population. They agreed to work with us in this pilot study. So now we'll test in the phase 1 around 50,000 couple every year with this expanded premarital screening and counseling program. And why we do this? We're not only testing them and leaving them alone. We're offering them, if they want to proceed with this marriage, we cannot prevent them. But here you have the choice of doing the pre-implementation genetic diagnosis, which means IVF minus the bad gene. Or if you exactly know what the mutation, you even can do the prenatal testing for this target or getting from the cell-free fetal DNA. Or do you have the opportunity now to expand your newborn screening, so you don't have to wait till the kid is 1 or 2 or 10 years, you know what's going on. And the circle goes through. So this program will hopefully kickstart in January of 2019, and it will be on the national scale. And this is what we've been doing for this program so far. I told you. So it covered almost 3,000 OMM disorders, and we validated 50,000 archival samples, and the call rate was around 99.7%. So the idea here is to have it as genetic matching, if you will. So the idea is, even from high school, even in college, you can do this test and have information. And when the time comes you want to marry or you want to have a partner, he will do it. If he didn't do it or she didn't do it, they can do it. But you have the option to do it very early on, even before getting into any kind of commitment. And you will have this information available to you, so you will have a choice when you want to proceed. How do you want to proceed? The current capacity is to do 100,000 persons per year, 150,000 couple per year. This is the number of actual marriages in Saudi Arabia every year. Sorry if it was in all over the places, but when I asked Sarah, what do you want me to talk about? She said, a little bit of Saudi Genome, a little bit about you, so I hope I covered both in this. And there is tons of people working on this. I just joined them. I was honored to join them. And actually it means a lot to me in person. 30 years ago, we did not even have anything. And going back now, working on a national program, trying to make more, to help more families, it's actually very wonderful. So there is many PIs, researchers. As I told you, over 80 PIs and 100 of researchers. There is the team in bioinformatics, biomedical informatics, which really I don't think this work can be done without all the help they gave us. And also the IT infrastructure, and of course all the families and patients and even normal volunteers who gave us the sample to them. And again, just leave you with this quote from King Salman, who said, the ultimate goal is to save lives of the future generation. Thank you very much.